Transcription in prokaryotes begins with template binding. This is where RNA polymerase bonds to the promoter region right before initiation. So, to begin with, RNA polymerase creates phosphodiester bonds between ribonucleotide triphosphates. These are shown here because they have an OH group at the 'two' region, and that also represents RNA. So, RNA polymerase cleaves this part off, and these bind always to the 3' region. They always bind to this 3' region. So for RNA polymerase to bond to the promoter region, we start off with the core enzyme, but that needs a sigma factor. So the sigma factor comes in, bonds to this, and it creates the hollow enzyme. The hollow enzyme is the whole unit, including the sigma subunit. So this bonds to the -35 and the -10 consensus sequences and regions of the promoter. So for elongation to occur, RNA polymerase, the core unit that has a sigma factor, needs to get rid of the sigma factor. So the sigma factor goes away, and RNA polymerase is able to elongate, and then is also able to terminate. So RNA polymerase reads the template strand down here of DNA, 3' to 5', and it mates RNA 5' to 3'. This top strand is the coding strand, and that's the non-template strand, and that is also seen 5' to 3'. Termination can be shown with inverted repeats. This is where RNA is being made, and you'll see a sequence of RNA here of the ribonucleotide triphosphates, and then later down you'll see an inversion sequence of that same thing. So this creates a hairpin loop, and these all bind, and later down in DNA you'll see a many adenines here, and then RNA polymerase makes many uracils right here. When these bond, they're very, very weak, so that just throws off this RNA unit, and termination happens. So if the -10 box is deleted from a gene that codes for an enzyme to metabolize sugar, that might not allow the sigma subunit to bond to each consensus sequence in the promoter. It will be able to bond to the -30 or 5 region, so that could allow RNA polymerase to see the promoter region and initiate transcription. But it may not also, because it doesn't have each one that it needs. So sugar being metabolized might not happen. These are the references.